Hi everyone, my group will be presenting on Clustal W and FASTA sequence. The members of my group are Yukita Budram, Christian Bacchus, Jamie Singh, Ravindra Jodho, Fonda Austin, Trevor Tobin, and myself, Louisiana Diru. I'm going straight into our presentation. I will firstly be looking at what exactly is Clustal W. So basically, Clustal W is a general-purpose multiple sequence alignment program that is usually used for aligning protein or even nucleotide sequences. It can produce biologically meaningful multiple sequence alignment of divergent sequences. It can also calculate the best match for the selected sequences and align those matches in order for their identities, similarities, and their differences. Now, steps in Clustal W. So firstly, what is done in this program is that it calculates all the possible pairwise alignments, and it would then record the score for each of those pairs. It would then calculate a guide tree based on the pairwise distances, and this is known as neighbor joining. Thirdly, it would find the two most closely related sequences, and then it would align these sequences by the use of the progressive method. And now, what exactly is the progressive method? What exactly happens in the progressive method is firstly, it would calculate the consensus of this alignment. It would then replace the two sequences with the consensus, and it would find the two next most closely related sequences, and these sequences could be a previously determined consensus, and it would then iterate until all sequences have been aligned. Moving on to the next step, step 5, what happens in this step is that it would expand the consensus sequences with the gapped or original sequences, and then it would report the multiple sequence alignment. Now here I have an image that is showing the steps in Clustal W algorithm, and we can also see in the middle here what exactly it would look like at each step. So for the first step here, we have pairwise alignment, and this is where they would calculate the distance matrix. And this is exactly what the distance matrix would look like for the various sequences. Then they would use that distance matrix and they would calculate the or create the unrooted neighbor joining tree. And this is what that would look like. Next, they would create the rooted neighbor joining tree by using sequence weights. And these numbers here are what are the sequence weights. These are the weights for each of these sequences. And the next step is the progressive alignment, which is done with the use of the guide tree. So in this step is basically where all the sequences will be aligned to form the multiple sequence alignment. Moving on, I have an image here showing exactly an example of Clustal W alignment. And in this specific example, this is a Clustal W alignment of an amino acid sequence between S. epidermidis and S. aureus. And here we can see that these sequences that have this in front of it is the S. epidermidis, and this one is S. aureus. And these various colors that can be seen in the picture, they represent this different aspects of this amino acid sequence. So basically, this is what would be outputted from the Clustal W program when trying to figure out the multiple sequence alignment for organisms. I will now hand you over to my colleague who will continue with this presentation.
Good afternoon, everyone. I'll be taking you through what is the FASTA sequence. Now, the FASTA sequence is a pairwise sequence alignment tool which takes input as nucleotide or protein sequences and it compare them with existing databases. Now, when searching the whole database for matches to give a for a given query, we compare the query using the FASTA algorithm. And so the FASTA compares a query string against a single text string. And so this is a text-based format and it represents either a nucleotide sequence or a peptide sequence. And for DNA and proteins, it is represented in one letter IUPAC nucleotide code, or you have the amino acid codes. We'll get down to that later in the presentation. Now, the FASTA, it produces a local alignment score for the comparison of query sequence to every sequence in the database. Now, the FASTA file description, it starts with the greater than sign. And so this indicates that the start of another sequence. So the file, since it usually starts with a greater than sign, the first line in the sequence it's called a description line, which may hold a descriptive information about the sequence in the lines, and it also has the name of the gene selected. Now, in each row, there will be 60 nucleotide or amino acids only. And so the FASTA sequencing, it is used to find the local similarity between the sequences, and it also calculates statistical significance of matches. Also, the FASTA sequence is used to find a functional and evolutionary relationship between different sequences. Now, as you can see here, we have 3 sequences. Now, in this FASTA format sequencing format, as you can see, each line begins with a greater than sign. Also, in the first line, we have the descriptive ID, which basically tell us what sequence we're looking at. As you can see here, we have part of a caspid of the CR asphage. We have the E. coli, which is the hypothetical protein. Then we have part of the caspid CR asphage again, but each has different numbers, which basically shows that they're both different something. Now, looking at the letters or codons. Now, each code, it represents one of the nucleic acid. Now we know T for thymine, A for adenine, C for cytosine, and G for guanine. And so this is basically a nucleic acid FASTA sequence format. Now here we have another FASTA sequence format. And as you see, the first line begins with a greater than sign, and it contains the name of the sequence. And in this sequence, it's an amino acid sequence. How do we know? Because it has different letters, and each letter represents a different amino acid. Now, this is a key that basically shows what each letter in the amino acid codes represent. And this here shows what each letter of the nucleic acid code represents. Now I'll hand you over to the next presenter.
Yes, so good afternoon, everyone. My name is Von Losted, and I'll be taking you through the creation of a phylogenetic tree using the results of Clustal W, Now, firstly, a quick recap on what is a phylogenetic tree is basically a diagram which helps to show the phylogeny or the evolutionary history of an organism using the different branches with, with the length being proportional to the amount of evolutionary diversity. So, the first step that you'd want to do when trying to construct this phylogenetic tree using Clustal W is to identify the sequence or the query sequences that you want to align, such as the sequences of basically a bacteria and the human and so on. After which you can then copy the sequences, the fast sequences for that organism or that protein, and then paste it into Clustal W, or you can compile all of them into one document and then place them into Clustal W using the choose file option. Now, the first step, similarly to the other informatics tool, you'd have to visit the Clustal W site, where you would then be given a option or a display option, as you can see here. And what you'd want to look for is the little icon that says phylogeny to your left. After you click in on that, you would then be given a screen which asks you to launch Clustal W2. You would then click OK or click on the icon, which would then generate you a screen similar to this. Now, after you have entered Clustal W2, what you'd need to do is import your sequence, as you can see here with my choose file option, or as I said, you can paste them in one by one or one at a time. Now, the difference is with Clustal W2 is that the description line or the force line that you see does not require a greater than sign, but it does require the phase Clustal in it, which means you must put in the word Clustal and then the name of your sequences, or else it would not work. After you've done that and then pasted your sequences, whether it be singularly or with the choose file option, you can then move on to the parameters or the various options below in step 2, where you then have the tree format. The format that you'll be looking for is the one that says default. There are also other formats, but we're not looking at that today. And then there are other options such as distance correction and exclude gaps. Now you'd want to leave these features on and off respectively, simply because if the distance are off or the or excluding the gap feature is on, it may create a phylogenetic tree that is less accurate, and we don't want that. The final feature that the final two features are the neighboring joining feature and the PIM. Now you'd want to leave the PIM off simply because PIM is the percent identity matrix, which basically shows the percentage of relation or similarity between the different sequences. And with creating a phylogenetic tree, this feature is not really applicable. And with that, you then move on to your third step, which would be to either allow the program to notify you when via email when the program is completed the task. Now, there's also an alternative way of creating a phylogenetic tree from Clustal W. And that is to say, if you've already run Clustal W and found the sequences and so on and already aligned your sequences, and then later on you decided to create a phylogenetic tree, there's always an option at your end result which says send to Clustal W2 phylogeny. Now, upon clicking on that, you would then go through back the whole process of creating a phylogenetic tree very simply. You would then be generated this output here that you can see, where I have my phylogenetic tree at the bottom, and at the top you may see something that is you may call gibberish, but it is actually computer language for the phylogenetic tree that you see below here. The ones and eights and the different commands such as ref and so on are basically what your computer uses to understand and generate the phylogenetic tree that you have here. You can also download this file or the file above and then transfer it to another computer and it would work similarly. Additionally, you may have other features which can show the distance or hide the distance between your sequences, or you can transpose the diagram and create a cladogram from it instead. And with that, I would to sum up everything that we've went through. Now, Clustal W is basically a 5-step, simple multi-sequencing program which is used to align nucleotide or protein sequences. It can also be used to generate physical graphs which can show the relationship between these different sequences. And we went through that as a phylogenetic tree. And lastly, we also went through the FAST sequencing program, which, like the Clustal W, is used for sequencing nucleotide and protein sequences. But the key difference is that Clustal W is used to compare sequences that you choose, meaning that you would copy the two sequences that are being compared to each other, whereas FASTA basically compares the sequence that you give them to a larger database to determine whether the sequence that you have has already been identified, or to tell you exactly what sequence you have if you do not know what it is already. And with that, I will then come to the conclusion of our presentation. And I'd like to thank you for your time. And these are our references.